Hi students, in this video you are going to practice calculating the biodiversity of an intertidal zone using a quadrat. Um, for this assignment, you can just come onto Canvas and click through the beginning directions. You do need a spreadsheet, and you can click here and create one, and it'll create something like this, which is a Google Sheet that you can modify. Usually there's also one in your lab manual, and I'm always fine with you writing things on your own paper if you want to record things that way. But once you go and click through the assignment, there are first some directions on how you can estimate the coverage of a certain um, type of animal, or you can do a direct count of an animal. So we'll see in here that there's some things like algae covering a space where it's covering maybe half the space. So instead of calculating the algae as one, you could just say the same algae plant, you could just say it's covering 50% of the screen or something like that. So sometimes a percentage makes sense, or sometimes you'll look at a picture and it has like 7 hermit crabs, and it's better to say 7 hermit crabs than say like 3% of the quadrat has hermit crabs. It's better just to count the exact number. So let me show you some of the organisms that you'll see just in in this instance and in this practice lab that you're going to be doing. So in this practice lab, they have some live barnacles. These are barnacles that have the trap door still in function inside. And if you haven't seen barnacles in real life, they do have this outer shell, but then this other part inside is like a trap door that opens and closes and lets like the feather-like organism that lives inside to come out, and it filters water as it passes by. It's kind of pretty. Um, these are dead barnacles because you can see the trapdoor is chipped out, meaning the living thing is dead. This is a green hair algae. Here's some live mussels and some sea lettuce next to it, and brown algae. And then in this, some limpets. So it's going to be pretty basic in these pictures that you're seeing. I'll tell you that if we do this as a live thing down at the beach in this course, you will end up seeing in Southern California way, way, way more diversity in this. This is really kind of a light version or a very basic beach. Um, this is what you see in this sample here. So these are uncomplicated samples. So this is a very basic version is what you're seeing here. Okay, so here I actually zoomed in on these slides because you can see this as is a Google slideshow. So I'm just going to come over here to the slideshow version, and I'm going to zoom in here. Okay, so this person has a tiny quadrat. It's very tiny, maybe 6 inches across. And you can look in here at their first place that they laid their quadrat, and there is nothing living inside that I can see. So I'm going to go to my like spreadsheet, my data sheet, and the first thing I'm going to do is write down a description of the photo, and in this one I'm just going to write that it was like sandy. Okay. And then barnacles, I'm getting 0, live or dead, no limpets, no mussels, no green hair algae, and nothing miscellaneous. That was just really basic. Okay, now I'm going to go to the next one. Okay, so in this one here, I am seeing when I am zooming in, there are a lot of barnacles. And you can see that most of them are no longer alive barnacles. I am seeing dead. Now I'm seeing like one here that might be alive right here that I can see the trapdoor. I think I can see the trapdoor on 2 of them here. And you might pick up a few more, and if you do, that's okay. I may be going a bit fast. So I got 1, 2. What happened to that other one? I saw 3. I thought I saw a couple more. 4, 5, 5, maybe 6. I can't tell for sure. That could just be full of sand. I'm going to say there's 6 that are alive. And see, I was able to count them. So on my spreadsheet, then I'm going to go sample two. I'm just going to call this one barnacles, and rock. Okay. And you can name it whatever you want. But barnacles alive, I'm going to say I found six. Okay. But barnacles dead, I want you to look at it a different way. It would be very, very, very hard to count every single dead barnacle. So another way you can do that instead of counting them individually is you can also just do a percentage. Like if we scooted all of these dead barnacles into one like corner, what percentage of the whole thing would it take up? Okay, so for me, I kind of visually break things down. So if I had like string going across here and going down here and here, I feel like this is almost one of those boxes, like maybe like 1/9, maybe a little less than a ninth, maybe a tenth. So for me, I'm going to write that the barnacles, the dead barnacles, were 10% cover. 10% cover. Okay. Then it says limpets. I saw going back. Let's look at it again. And one more time, if you need a limpet, this should be a little purple guy, like with radiating beams is our limpet. I don't see any of those. Okay. And so I'm going to write 0 on my spreadsheet. The mussels are like the little like clam like guys, but they're going to be very small in this. And I saw 0 of that. I didn't see any algae, but let me go check again. No algae. That would be green. And I didn't see brown algae either. Okay. So now I'm going to go on to my third quadrat. Okay. And now in this quadrat, I can see some more potentially dead barnacles. Okay. And now you know what those look like. I don't see any of them that are alive yet. And I see a couple limpets. And they are few enough in number that I could just count them. Okay. And do I see any mussels? No, but I see some more dead barnacles here. A couple of them. Some of these are dead barnacles. So I am going to count those up. I think I saw 3 limpets. Let me see if I see mussels in there. I'm going to scroll ahead and see. Oh, more limpets and barnacles. Let's see when we get to another one. Okay. See these little black, darker brown blacks, maybe, yeah, in there? These are going to be mussels. So when I go back to number 3 that I'm on, and it's okay to compare to other ones so that you can get it calibrated, I am seeing a couple mussels, okay? Like 3. And I see 3 little limpets, if I can count this guy. I'm going to count it. 1, 2, 3. And then a lot of dead barnacles. So when I go to my sheet, I'm going to record the limpets, the mussels, and the dead barnacles. Okay. And the dead barnacles, I'm going to estimate a percentage of cover. And I'm not going to tell you what I would put because I want you to come up with an answer. Okay. So there's number 4. I'm not going to help you on that one because I think you can do it. 5. Here's 6. I'm seeing some mussels. I see limpets, but these guys were outside of the quadrat. So you're going to have to figure that one out. But this is the first one that I see some green algae. Okay. So for this green algae that is here, again, I want you to do, you could say that this is just one big green algae plant, but it's more descriptive to say that it is taking up a certain percentage of this. And so again, if I was to cut this in half, it's definitely less than half. So to me, it looks like it's more like a third of green algae is in here. So that's how I would estimate that one. You are going to fill out your samples and write how many in each space, describing it as you move along. And you're going to practice doing these quadrats. You also have a different sample site. So here's sample site 2. and they did it again. And so you're going to go through and do that. That's how you get sample location 2 and all the samples, and then location 3. So I'll maybe do one more. Let me get you started on location 3. Let's go see what's going on there. So here's location 3. That's kind of cool. They went across the rocks and out straight towards the water. So let's do one of the middle ones here. This one looks pretty interesting. Okay. So here, just looking at it first, I see definitely some green algae, but then why is this green algae like so odd looking? It's not like straight and feathery. And then I have this clearly brown algae. Okay, I'm going to go back to my ID, species ID, and way zoomed in on here was the sea lettuce. Okay, so sea lettuce is this, and it looks very different than the straight um, green hair algae. So one looks like hair, and the other one kind of does look like sea lettuce. Okay, so when you go and count up this space, make sure you do, I would do a percent cover with the green sea hair on this, and I would do a percent cover with the brown algae, but I might just count the individual sea lettuce plants, or you could just picture putting them all in just a tiny little space and do a percent cover of the sea lettuce. I would be fine with either one. Don't forget these little guys down here. They look like sea lettuce too, and one hidden over there. So I could see making a case of doing either counting each plant, or you could do a percentage of cover in that. So when you turn in this assignment, you're going to come back to Canvas. You are going to answer some questions in here, like what are 2 abiotic factors that regulate carrying capacity of mussels in location 1 and 2? Okay, so we didn't talk about that here, but you can look up a little more information about carrying capacity. But I'll tell you, abiotic factors are what non-living things are affecting the mussel population. And you should look back at the original picture. This is sample sites site 2, and here was sample site 1 here going across the rocks. And as they got further and closer down to the beach, this is when they're starting way far back here in this first picture, and then their second picture they started to get up on the rocks, and then they got closer and closer and closer to the water. And you can see as they got closer to the water, they got more mussels. So that question is, what abiotic factors, things that aren't alive, are affecting how mussels are growing? So you could talk about the effects that, abiotic factors would have, like the sun, the water, the salinity, the pH, the amount of nutrition that is in the water, things like that is what you should mention. And then look at live and dead barnacles in all 3 samples. What ecological tolerance factor seems to regulate where you see barnacles? So what is kind of regulating where you see barnacles? Where did you see barnacles the most that were alive, and where did you see barnacles that were dead? And what's kind of the difference? What factor is helping them to either live or die? And then look at your samples again. What factor seems to influence the algae growth? What has to be there for the algae to grow? And believe it or not, that answer should be maybe a little bit different than for the barnacles. And then examine the population of limpets. Are limpets, it's asking, are they a generalist species or a specialist species? Now, a generalist species is a species that'll generally eat just about anything. And a specialist species is like, we're really picky. We do not eat very many things at all. If I don't have at least, this one food source, I'm going to die. So, do you feel like limpets are generalist or a specialist? And you explain why. And then examine the population of limpets. What adaptations might they possess that could explain the distribution in each of the three sample sites? So, what do limpets have that make them better able to survive, or that makes them able to survive in multiple locations because they were able to? Um, then which sample site had the highest diversity? Like, so you're going to go back and you're going to see which one had the most different types of organisms or a lot of growth in their different spaces. And then write down why you think there was a lot of diversity there. For the last one, you're going to upload an image of your data sheet. You're going to turn that in so that I can give you a grade for it. Please make sure that all of your answers, you'll notice they're worth like 10 points each. Make sure that your answers are thorough and college level.